Welcome everyone to our next webinar series. And today it is my greatest pleasure to introduce our speaker, Chris Parker. Chris earned his PhD in chemistry from Yale University under the supervision of David Spiegel. And even during his PhD, he was already working on bifunctional molecules. So he was recruiting the endogenous antibodies to a specific target, resulting in the immune-mediated clearance. Afterwards, he carried out postdoctoral studies under the supervision of Ben Cravat, where he developed new chemical proteomics methods for global ligand and target discovery. Chris started his independent lab at the Scripps in 2018, where he develops chemistry-centric strategies to investigate human biology and in therapeutic context. Thanks so very much, Chris, for joining today, and we are very much looking to your seminar.
Perfect. Thank you so much for the introduction and thank you so much for the invite. I'm such a fan of this webinar series. I think I've seen every episode, and so it's a really great privilege to be able to present some of our lab's work. As sort of mentioned in the introduction, our lab is broadly interested in developing chemical probes to study protein function, usually in the context of diseases like immune conditions and cancer. And to enable us to do that, we rely quite heavily on the use of mass spec-based proteomics to allow us to sort of map small molecule protein interactions and modifications and obviously changes in abundances, ideally in relevant contexts. And so I'd say there's sort of three major areas of our lab. The bread and butter of what we do is chemoproteomic method development. Here we try to innovate strategies and tools to globally map the chemical tractability of the proteome directly in live cells, mainly through the use of photoaffinity-based strategies. And the goal is to use these methods to pharmacologically access proteins that have compelling value to human health and disease roles, but have presented challenges using conventional chemical probe discovery strategies. And so we do quite a bit of target ID as well of bioactive molecules like screening hits or metabolites, etc. And the third area, which is a little bit newer to us, is focused on proximity-inducing molecules and methods to study protein-protein interactions. And here we try to leverage a lot of the learnings from our chemoproteomic ligandability studies to develop molecules that can redirect protein function. We're of course, like many folks here, interested in targeted protein degradation, which I'll share a little bit later on today, and thinking about ways to redirect other alternative post-translational modifications like protein acetylation, which I won't talk about today. But I plan on sharing two degradation stories. One, I would say, is a very sort of serendipitous discovery emanating from our ligandability studies, while the second is a little bit more intentional and is really focused on expanding the scope of targeted protein degradation. And so first, just a little bit of contextualization to set the scene of why we do what we do. There's around 600 human proteins targeted by about 1,400 clinically approved small molecule drugs. And depending on what resource you look at, there's anywhere between 3,000 and 6,000 disease-related proteins. This number is based on extensions from currently drug protein families and things like genetic screens. So these are proteins that have clear roles in human pathology, disease pathology, and have evidence of being liganable, but sort of lack advanced chemical probes. And this, of course, is a small fraction of the entire proteome encoded by the genome, 20,000 proteins, which is sort of an underestimate also when you consider proteoforms emanating things from like alternative splicing and post-translational modifications, which are really important, obviously, to protein structure and function, but also the way a small molecule might interact with a protein, and really is a main reason why we try to do most of our studies directly in live cells. And I show these numbers more to sort of highlight the gaps. There's a variety of reasons for these gaps. Obviously, in exploring enough biologically relevant chemical space to find a molecule to do what you want it to do. A lot of proteins, even if they have a clear disease role, might not be compatible to conventional sort of in vitro based high throughput screening methods, and they really need their native environment to be functionally and structurally relevant. A lot of proteins, again, if they have those clear genetic links, lack direct proximal readouts, making it challenging to develop a chemical probe to perturb those functions if they're not really well established. And of course, I think this definition is dwindling, but there's whole classes of proteins that are sort of thought to be intractable in small molecules, although that I think view is slowly fading away. And so I'd argue the field of chemical proteomics is really well positioned to tackle many of these challenges. And the field really is based on the use of probes that profile and report back on various features of proteins, such as enzymatic activity, amino acid reactivity, or just plain old ligandability, or the ability to bind small molecules on a proteome-wide scale. And the field as we know it, was founded on the activity-based protein profiling, which was pioneered by my postdoc advisor, now colleague here at Scripps, Ben Cravat, as well as Matt Boggio at Stanford. And so the general principle here is that these are active site-directed probes that mechanistically react with related classes of enzymes and are used to monitor not only enzymatic activity, but occupancy of active sites. So they can be used as reporters for binding site occupancy and be used to find ligands or inhibitors of these enzymes. And so, although this sort of initially started out with serine hydrolases, it's been expanded into nearly all classes of enzymes. And more recently, this sort of general approach would be extended to enable assessment of small molecule binding beyond active sites by targeting nucleophilic side chains with electrophiles. And this really opened up the floodgates, I'd say, for chemical probe discovery on a proteome-wide scale. Once again, the methods were initially pioneered by Ben Cravat's lab through the targeting of cysteine residues. But now we have ways to monitor covalent engagement of lysines and tyrosines and methionines and many more. And what I think has been really impressive, even though these are relatively new technologies, there's been a number of biotech companies that were founded on these general platforms that already have, within less than a decade and sometimes less than 5 years, clinical candidates that were born out of these platforms, which I think is extremely compelling and speaks to the utility of these platforms to find first-in-class small molecule modulators. And so, although these methods are quite powerful, they're obviously limited to a subset of the proteome. Either they require enzymatic activity or sort of this conveniently placed nucleophile within a binding pocket. And so our lab, I would say, aims to explore whether or not these general principles can be extended to, in theory, all proteins and all binding pockets independent of activity or the presence of these nucleophilic residues. And so there's really no universal probe or electrophile for us to probe all potential binding pockets. And so instead, we utilize what we call fully functionalized probes, which are really just drug-like ligands attached to a photoaffinity handle. And we deploy them in cells, tissues, or lysates. And so you have this reversible interaction, and you shine light on it. And then you could do click chemistry developed by my other colleague, two-time Nobel laureate Barry Sharpless, to enrich those proteins and identify and quantify, et cetera, by mass spec. And in doing so, we kind of do this over and over again, and we build up a protein small molecule interactome map where we try to sort of broadly identify proteins and sites on proteins capable of accommodating drug-like small molecules. And then we advance this information, ideally, into more elaborated compounds that can actually modulate protein function. So we have an ever-expanding library of these fully functionalized probes in our lab. Each member contains this diazurine and an alkyne click handle. We've played around with different variations, and we and others, including Christina Wu at Harvard and Joe Fox's lab at UD, have explored diazurine reactivity, and we sort of understand the best tags to use for photoaffinity approaches. And our original library is actually starting this work when I was a postdoc, composed of what we call scout fragments, and these are just promiscuous ligands that really the goal is to try to find small molecule binding pockets that are ligandable in cells, and so that we integrated principles of fragment-based drug discovery to screen smaller libraries that are more promiscuous, and then elaborate upon them to make more potent and selective inhibitors. But these compounds are often too promiscuous, making it challenging to decipher authentic interactions. And so we subsequently found that you can integrate these libraries with stereochemical elements to really expedite discovery of authentic interactions. Due to the fact that if it's a stereoselective interaction, enantiomeric probes or stereomatch probes have similar physicochemical properties and generally sort of similar proteomic, non-specific proteomic interactions. And so if it comes off as being stereoselective, it's often driven by molecular recognition and not some other physicochemical event. More recently, we've sort of expanded this into more drug-like space, being inspired by combinatorial chemistry, natural product chemistry, and even exploring other forms of chirality. But I really won't get into too much more of library design, but mainly to say we have several thousand members, and and we have different chemotypes depending on the objective. And so this is a bit of what our data might look like. I'm showing here a sample heat map of observed small molecule protein interaction. Probes are on the x-axis, proteins are on the y-axis. Dark red indicates a target that's hit quite a bit, light red and white less so. One of the main takeaways here is really just to emphasize that such analysis allows us to broadly assess interactions both from the perspective of the protein. So you could see a very promiscuous proteins that seem to interact with everything oriented at the top of the heat map, as well as from the probe. For example, probe 1 seems to be quite promiscuous, interacting with many proteins. Probably not a good lead for developing ligands for any particular protein, but as you venture to the right-hand lower part of the heat map, you start getting a little bit more protein-wide selectivity and SAR. Over the past few years, we've identified ligands for about 8,000 proteins, human proteins. We have pretty strict standards on what we consider a ligand. They span diverse functional classes. Of course, we had enzymes, but we also hit about every other class of functional class of proteins, including transcription factors, adapter proteins, a lot of UPS machinery, and scaffolding proteins and things like that. We can also map the site of interaction of probes on proteins and cells using a slightly modified workflow. So we do our treatment with cells. We chop up the cells with the protease and elute away the tryptic peptides. And what's stuck to the resin is the probe modified peptide. And using sort of an orthogonal linker, you can release those probe labeled peptides. And the ability to robustly map the sites of adducted sites of photoaffinity probes has been sort of historically a challenge, mainly because when you shine light on diazarines, they form this reactive intermediate called a carbene, as well as other intermediates that in theory you can insert anywhere on a protein, side chain, backbone, between any bond. And so this creates a number of issues. One, you sort of have this massive search space when you do the proteomics, in contrast to, let's say, covalent probes, where it's reacting primarily with cysteine. We have to tell the search algorithm, look for a modification anywhere on any protein at any amino acid in any bond. So this creates a search space issue. In addition, probes, you often have multiple adducts within the same tryptic peptide, and they have often the same retention time, are isolated roughly the same time, and so this gives rise to something called chimeric spectra, where you have different peptide sequences that are embedded within the spectrum, but most search engines often just assign one sequence to one spectrum, and all the other spectral information is kind of lost. And you also have issues like probe fragmentation, which can complicate the readouts. A talented postdoc in my lab, Jacob Wozniak, developed a workaround solution to this. I really won't go into details, but you can check it out at this paper we published last year. We call this new workflow diazarine probe labeled peptide discoverer, or DISCO. Main takeaway is that it increases we sort of took into account these spectral issues, and we're able to develop a predictive model to assign whether or not a spectra contains a probe labeled peptide or not. And this increased not only our coverage, but also the site level resolution. So we can, with greater confidence, identify probe labeled sites on proteins directly in live cells with endogenous proteins. And collectively, our lab has identified about 7,000 binding pockets through this approach. And this allows us to do quite a bit of meta-analyses. And so we find that they often occur near functional sites, allosteric sites, near sites of post-translational modifications, and largely occur near predicted pockets. So we can utilize either PDB structures or predicted structures to do this sort of analysis. And what's great is, we can integrate this with multiplexing or label-free based quantitative proteomics, and actually get essentially KDs of these binding events, both of the probes, but also in competition with site level resolution, which becomes really powerful when you're trying to figure out exactly where your probe is hanging out, or when you're trying to optimize a tagless analog. And we could also integrate this with molecular docking to sort of predict orientation, which could guide SAR and also give us confidence when we identify new binding pockets. For example, I've shown an example here of a newly identified pocket on this GDI2, which is involved in sort of RAS signaling. We bind this lipid binding domain. One other thing I wanted to highlight is we often come across what we call neopockets, which are pockets that form at the interface of protein complexes. And I'm showing an example here where we found a labeled site on the CDK1, and when we tried to validate this interaction on recombinant protein, we weren't able to, but when we do it in cells or we added other components, we're able to see reconstitute binding. And once we did the modeling and site mapping, we see that we're binding at this pocket. And we come across this all the time, which I think really points to the value of doing ligand discovery in cells rather than in isolated contexts. And so once we have all this information, what do we actually do with it? Um, uh, as I sort of alluded to before, the probes can serve as readouts of binding site occupancy via competitive displacement. And so we can either elaborate and sort of brute force medchem the initial probe hit, or use it as a pocket probe and do a counter screen with tagless analogs to find a more potent selective compound. And I'll share one story that will get to the topic of today around targeted protein degradation. So as I alluded to before, our lab is interested in autoimmunity, and there's a number of autoimmune disorders. But the sort of main takeaway is most of autoimmunity is caused by the unchecked activation of components of both your adapted and innate immune response, either due to environmental or genetic factors, usually a combination of both, resulting in tissue damage. But really, it's often instances of your innate immune response plays a key role in activating, perpetuating these responses. And mainstay treatments are often things like NSAIDs or steroids, which reduce pain and inflammation, but could have harmful side effects if taken chronically, or broad immunosuppressants. Biologics have an increasing therapeutic presence, although they're expensive and considerable portions of the population are still sort of unresponsive. And so there's really a need to develop new small molecule therapies that target the main perpetrators of inflammation, often components of your innate immune response. And so if we take a closer look under autoimmune conditions, immune sensors, which are typically dedicated to the detection of infection, are sometimes usurped and initiate and propagate autoimmune disease, and specifically the unchecked activation of endolysomal toll-like receptors, TLR 7, 8, or 9, which are activated both by viral nucleic acids, but also self-nucleic acids, are main events in autoimmune disease, including things like lupus and psoriasis. And so these TLRs get activated, go on to activate NF-kappa B and IRF signaling pathways, producing pro-inflammatory cytokines like type 1 interferons and TNF alpha, for example. And so it turns out that endolysomal TLR signaling is completely dependent upon this multipass transmembrane protein called SLC15A4, which was initially sort of discovered from a forward genetic screen from a former Scripps faculty, Bruce Beutler. Basically, they found that you knock out SLC15A4, you don't get endolysomal TLR mediated production of pro-inflammatory cytokines, and another immunologist at Scripps, R.A. Theophilopoulos, later and followed up by many others in the field, basically showed that animals that don't express 15A4 are sort of protected against autoimmune disease like psoriasis and lupus and IBD, for example. And there's actually really strong human genetic correlation between mutations and SNPs and 15A4 and autoimmune disease. And so the sort of perfect scenario of biology here that points towards the development of inhibitor of SLC15A4. But at the time, there was no structural information. No one really knew what it did. It's a member of a larger class of proteins called SLCs, which are transporters, although it was sort of shown that transport function doesn't have anything to do with its TLR function. And at the time, there's no chemical matter. And so if we want to make an inhibitor for a protein that has no proximal readout, how could one do that? It has 12 transmembrane domains. It's heavily glycosylated, so we wanted to do this in cells. And so we decided to take an integrated approach where we screen for probes that bind to SLC15A4 directly in primary patient immune cells that also suppress this downstream phenotype, that's the production of pro-inflammatory cytokines. And I really won't go through the details, but this plot just kind of shows the enrichment of different probes for SLC15A4. So we found a bunch of probes that hit SLC15A4 that are not functional in terms of downstream phenotype. And we found a few that do. One of our top one is this probe called FF21. This is a structure. It's a 2-aminobenzimidazole. We showed that we can remove the tag and block probe labeling, which is important. But the tagless analog in the downstream functional assay was pretty bad. Maybe 40 micromolar, probably even higher. And so we decided to launch what we call sort of a chemoproteomic guided optimization or medicinal chemistry And we made about 140 analogs using target engagement as a primary readout for optimization, as well as this downstream functional assay. One of our lead compounds, not our most potent, but one that we published on, is this compound called AJ230. We've made the probe version, showed that we're on 800 nanomolar to 1 micromolar IC50 in terms of cytokine suppression in primary human and mouse cells. And we always go back to the model system to ensure target engagement. So looking at active versus inactive enrichment and competition with tagless analogs, 1584 is consistently the top target of these compounds. The only known function of SLC 1584 at the time was this transport function of bacterial-derived glycopeptides, which end up being agonists for another set of immune sensors called NODs. So it transports, once bacteria are endocytosed, chops up the bacteria, these glycopeptides get shuttled out through SLC and are agonize these NOD receptors. So we just develop a reporter assay for that. And we show that our compounds block this only known function of SLC 1584 and quite selectively. So we can look at other subfamily library members and see that we see no inhibition of transport. At the time, there's no structures, but we were able to map the site. Subsequently, the past few years, there's been a number of structures. Unfortunately for us, it's the site that we see mapped is sort of on this unresolved loop in all structures, but we see the site adducted to a site that's very specific to SLC 1584 that's not present in other subfamily members, indicating to us, or consistent with the sort of selectivity we see in the transport assay. Long story short, working with our collaborator, John Tajaro, and his lab, we basically showed that our compound completely recapitulates the knockout phenotype in primary human and mouse cells. So we can stimulate endolysomal TLRs with small molecule agonists or with flu or COVID. You can stimulate NOD. Our compound blocks all of them. We didn't really know the mechanism at the time, so it was really important for us to find functional assays to show that we're selective. So you can stimulate with other TLRs, and our compound's inactive, so it's not a broad immunosuppressant. Since we were pretty confident this is the first bona fide SLC inhibitors, we wanted to test our hypothesis as to whether it could lower inflammation in immune cells derived from lupus patients. So these immune cells often have a heightened inflammatory state where they produce more autoantibodies and pro-inflammatory cytokines. So we can isolate those immune cells and just add our compound, and we suppress inflammation of producing those lupus patient samples. We did an initial sort of PD readout where you can inject mice with TLR agonists, and our compound, even though it's pretty lipophilic and has some issues in terms of metabolism, it's suitable enough to do in vivo studies, and we see suppression in vivo. And we're even currently pursuing efficacy studies in LPR lupus mouse models where these are very long-term, 20, 30-week type readouts, but we see very promising preclinical data showing that we're able to suppress manifestations of lupus like symptoms in these mice, which is quite exciting. At this point, we didn't really know the mechanism. When we do our chemoproteomics, we always use alternative methods to measure ligand engagement, such as CETSA, which uses heat gradient to monitor ligand-induced stabilization. Typically, a ligand stabilizes a protein. We see a profound destabilization that's specific to our active compounds, but not our inactive compounds. It's dose dependent. These kind of scary blots are because SLC is heavily glycosylated. If you add PNGAs and strip off glycosylation, you see coalescence of the band. Interestingly enough, when we incubate for long periods of time, after about 6 hours, 10 hours in primary cells, we start seeing protein loss. And we see this in every immune cell. We see it in human B cells, mouse B cells, different cell lines. And considering this is a endolysosomal transmembrane protein, the nearest cellular trash can, if you will, is the lysosome. And so we hypothesize that maybe our molecule is binding, somehow destabilizing the protein, causing lysosomal degradation. And so we add lysosome poisons like baphlomycin or hydroxychloroquine or ammonium chloride, which sort of neutralize the pH of the lysosome and disrupt the lysosomal machinery. And we see complete rescue of this degradation event, indicating to us that our compound is inducing lysosomal degradation. So we thought this was our mechanism, right? That our compound's binding and we're just degrading it through the lysosome, which is a really cool mechanism. And it even works in vivo. So you can inject these mice with our compound, or feed them our compound and then extract the B cells and see pretty substantial loss of 15A4, usually around 50–60%. So most 15A4 expressing cells in terms of lysosomal localization, about 50 or 60% is in the lysosome, the rest is in the ER or elsewhere in the cells. And what's really interesting is in the genetic studies, you only need a heterozygous deletion of 15A4, so 50% deletion to have the same sort of phenotypic effects and disease amelioration effects as you do with the homozygous. So depletion of 50% or more is great. We're excited. But a reviewer rightly pointed out that we actually saw disruption of signaling much faster time scale than the 14 hours or 10 hours it takes for degradation. And so we went back to the drawing board and hypothesized that this destabilization was a result of maybe disrupting a PPI. And so we used proximity labeling proteomics to map the interactome of SLC15A4 and found a lot of interactors belonging to mTOR signaling cascade. We went ahead and validated a number of those. We're kind of tapping into known biology around 15A4 at this point. It's known that if you knock out 15A4, you disrupt TLR-induced mTOR activation, and we see that with our compounds here. It's specific to endolysosomal TLRs, not other TLRs, and we even see this in vivo, telling us that we're probably on path here. We validated a number of these interactions. One of the top interactors is LAMPTOR1. LAMPTOR1 is part of a complex called a regulator complex, which sort of anchors the rest of the complex to the lysosomal membrane, allowing mTOR to be recruited to the lysosome. And so we hypothesized that maybe we're disrupting this interaction with our compound, and indeed we are in a very dose-dependent fashion. So we see disruption of LAMPTOR1, other components of regulator, LAMPTOR2, and maybe some ATPase subunits, which are also part of the mTOR signaling pathway, in a dose range where we see activity of the compound. And so this led us to a hypothesis that we're blocking TLR-induced mTOR localization, and indeed we are. So we can visualize this by microscopy in primary immune cells. And so we're tapping into some known immunology here. So the Polundran lab had shown that mTOR is essential for TLR-induced cytokine production by a pathway that involves the IRF5 and 7 activation. And so we basically showed that our compounds are blocking IRF5 and 7 phosphorylation, activation, and nuclear translocation in immune cells, sort of completing this story. And so, this first story, I think, is a really interesting tale of how mechanism-agnostic binding-first approaches can enable the development of first-in-class inhibitors for challenging targets like SLC15A4. In this instance, we found that our compound was actually disrupting a newly identified protein complex, which also led to destabilization and subsequent lysosomal degradation. So we don't know the precise mechanism of the lysosomal degradation yet, but it's, the lysosome has its own recycling pathways for destabilized proteins. And so it's probably getting some sort of endocytosis or clipping of the lysosomal membrane and then degradation through that pathway. And so I'm going to switch gears here now and share another story, which is a little bit less serendipitous. As I mentioned earlier, our lab is really interested in using chemical proteomics to develop ways to modulate PPIs and profile PPIs. We're, of course, interested in degradation and thinking of ways of making more proteins tractable to degradation, but also exploring, as I mentioned earlier, ways to redirect other PTMs like acetylation. We really like acetylation because it's unlike PROTACs or molecular glue degraders. Acetylation is often a gain of function signal or a change of function signal rather than being an inhibitory. And so we were really interested in the targeted acetylation in the context of transcriptional activation, or actually protein stabilization, because the acetylation occurs at lysines, the same site as ubiquitination. So you can imagine that if you block ubiquitination with targeted acetylation, you can stabilize proteins. And so we developed some tools to we call them ACE-TAG to explore targeted acetylation in cells. Won't really have much time to talk about that today, but I'll just direct you to a couple of these papers. But I rather get back to the topic of targeted protein degradation and share a newer story focused on ways to expand our understanding of sort of the chemically degradable proteome. And so PROTACs are don't need to be explained to this audience. They're, of course, heterobifunctional molecules that can induce proximity between E3 ligase and a protein of interest. facilitating its induced ubiquitination and proteasome degradation. And they've been now developed for a multitude of disease areas and have shown very promising results in clinical trials. Um, and so this is sort of just a somewhat up-to-date snapshot of some examples. But despite their potential, the vast majority of PROTACs really utilize pre-existing small molecule inhibitors, including all disclosed structures that are currently in clinical development. And I'm just highlighting a few structures, example of clinical candidate structures here, which are all based on previously established inhibitor scaffolds. And though these inhibitor scaffolds are obviously great starting points, they've been highly tuned and optimized and have great PK properties. They sort of face the challenge of retrofitting this finely tuned ligand with a linker and E3 moiety. Additionally, PROTACs ideally should confer some sort of advantage over the inhibitor that they're based off of. For example, the targeting perhaps scaffolding functions of an enzyme, or maybe increasing the selectivity of an inhibitor through this alternative pharmacology, or else it wouldn't be that useful. But I think perhaps the most obviously this reliance on well-developed ligands, or this trend of using well-developed ligands, inherently limits PROTAC development to a very restricted scope of targets that actually possess well-characterized and advanced ligands, which are often quite druggable targets that in areas that there's also a lot of competition. And so our lab, like many other chemoproteomics labs, has really spent a lot of time thinking about methods and chemical libraries suitable for proteome-wide ligand discovery. Chemical proteomics, of course, is basically a proteome-wide binding assay. And so we make these target agnostic libraries, like the fully functionalized probes that I showed, screen them, and go through this effort of finding selective binders, which may or may not impart any functional consequences on the protein. Often it takes a little bit of elbow grease to convert, an okay binder into an okay functional modulator, like the story I just went through. However, the screens themselves they're binding based, and we have a lot of other sort of really exciting binding based assays now available, DEL-based screens, for example. They don't really provide any information about function. It has to be inferred indirectly or somehow built in later. And so just sort of thinking about this as an alternative strategy, particularly in the context of degradation, we hypothesized that we could potentially make a different class of fully functionalized library members. This time, one focused not on binding, but just on function, ideally a function that we could read out by proteomics, since that's what we do. And so degradation is sort of fits the bill. And we rationalize we can borrow a lot of our learnings and principles from our proteome-wide ligand discovery studies to find binders and convert those learnings into libraries that can broadly survey proteins for their potential to be degraded, again, as read out by proteomics. And so, of course, a major concern is that by utilizing what is likely an unoptimized ligand, they would be too promiscuous or low affinity to actually facilitate protein degradation. And although we were obviously comforted that PROTACs are catalytic, that we might be able to overcome this low affinity issue through a catalytic mechanism, or perhaps the screen would just find highly cooperative binding events. Nonetheless, we were encouraged by sort of seminal work from the Cruz lab, as well as Nathaniel Gray's lab and Eric Fisher's lab, demonstrating that key factors that often associated with successful small molecule inhibition, so the binary complexes such as target affinity and engagement, don't necessarily predict high degradation efficiency or selectivity. And so they did a number of studies using a variety of different kinase inhibitors and showed that often really good kinase inhibitors do make really good degraders, but that's not always the case. And this gave us confidence that, if we did a screen where we're utilizing unoptimized binders, we might have the potential to identify degraders, even if the binders aren't that great. And so with this sort of thought process in mind, we designed and synthesized a library of 72 bifunctional molecules composed of a thalidomide-based cereblon ligand and conjugated through 4 different linkers to 18 small molecules. And so since these small molecules don't really have any identified targets, we call them target agnostic PROTACs or just agnotacs. And so for this library, we tried the best we can to focus as much on drug-like parameters, so the head groups sort of fall in the lower molecular weight. They're not too lipophilic. We really try to implement uh, uh, filters of diversity. And so we have similar diversity as drug bank, for example, and the diversity within the library, even though it's a small library, is quite good. We next employed TMT-based quantitative proteomics and screened this library of 72 agnotacs to measure changes in protein abundance relative to a control. I won't go through the details, but we explored various parameters such as incubation times, concentrations, and found that since these are probably not very well optimized compounds, we ended up getting more bang for our buck at higher concentrations. And so this kind of made sense to us since these are like fragment-like warheads. And so collectively, we identified quantified over 8,000 unique proteins, over 170 of which showed substantial decreases in abundance, greater than twofold over biological replicates. We noted that one of our compounds was really cytotoxic, and was a lot of the shifts that we saw in the proteomics were probably due to the cells dying. And so we kind of removed that from our number game, and collectively we identified 89 unique downregulated targets, or sorry, 60 unique targets, 89 total. And what was really exciting is that a significant portion of the targets fell out of what this sort of canonical druggable class. Most of them lacked available ligands. I think we only found one target that actually had a known degrader. The vast majority of them didn't have available ligands, and they once again span diverse functional class. Of course, enzymes, but also adapter proteins, transcription factors, etc. Obviously, proteins decreasing in abundance doesn't mean they're being degraded. We created a focus library that has sort of this degradation tag, a cereblon ligand, but it doesn't mean because we see proteomic changes that's caused by binding and degradation. So we pursued validation sort of using time course studies and dose responses, as well as sort of standard control experiments to confirm that the proteins being downregulated were doing so in a proteasome and CRL, colon ring ligase dependent fashion using MG132 proteasome inhibitor and the MLN 4924 netylation inhibitor. And we found that in most instances, our agnotacs display DC50s in the mid micromolar range and maximum degradation between 50 and 75%. Not super great potency, but again, these are often first-in-class ligands or first-in-class chemical probes for these proteins, relatively unoptimized. And for all the targets that we followed up, we see rescue with both proteasome inhibitor and netylation inhibition, suggesting that they're going through this mechanism. We certainly ran into instances that didn't behave through sort of the canonical mechanisms. So for example, ETS1 and TMEM-T05. ETS1 is a transcription factor. TMEM-T05 is a relatively uncharacterized transmembrane protein. We see that it's rescued with proteasome inhibition, but not netylation inhibition. And without going through all the details, we feel like with the number of controls and follow-up experiments, we demonstrated that this is probably just a ligand-induced degradation event through some other pathway. These other targets, including adapter proteins, UFD1, POD2, an enzyme, this transcription factor CHCHD2, and we found another BRD2 degrader if anyone needs one, are all being degraded through sort of these canonical mechanisms. Given that the vast majority of these targets would not only these be the first chemical tools, but the first PROTACs, we wanted to utilize several additional validation mechanisms to demonstrate cereblon dependence and target engagement. For example, we use sort of this methylated glutamide analogs, as well as VHL-based cereblon degraders to show that degradation is not only dependent upon binding to cereblon, but also the presence of cereblon. So, in each instance, when we use this methylated control, we see rescue, or if we degrade cereblon through this VHL sort of chimeric PROTAC, we see rescue of agnotac-induced degradation. We also utilize a number of tools to ensure target engagement, such as CETSA or photoaffinity labeling, our favorite tool, where you can make a photoaffinity probe out of the warhead, if you will, and do competition experiments with the agnotac showing that you are hitting the target that is being degraded. All this gives us confidence that all the targets that we followed up on are going through these sort of canonical mechanisms. I won't spend too much time on this aspect, but it's pretty much standard in the field. We purposely embedded a bit of linkerology in our library for different linkers. We had really no idea what linker would be optimal. And what we observed is that each linker generally led to the same number of degraded targets. There was no sort of preferred linker chemotype. And most targets were largely degraded by one linker type. And so this tells us that if we would have included even more linker designs, not only would we sort of be able to maybe modulate degradation of any given target, but would actually degrade newly identified targets without actually incorporating additional warheads. We also explored a little bit of optimization. So this was a UFD1 degrader. We expanded sort of our linker exploration as well as head group modification. This is just a sort of a little exploration showing that we can take the initial lead and increase the potency, 4 or 5-fold with relatively little optimization. But this again gives us confidence. SCR gives us confidence that we're on target here. We also dug a little bit more into several targets. In most instances, these agnotacts were the first available chemical probe, and so we wanted to use them to probe the targets a little bit more. As noted earlier, we identified a degrader for this protein called UFD1. Interestingly, this agnotac also equipotently degraded another protein called NPLOC4 or NPL4. Both are adapter proteins that form a complex with ATPase P97, also called VCP, which plays a pretty critical role in ER-associated protein degradation, or ERAD. Proteins that are misfolded in the ER are sent through this P97 ATPase, a process that's facilitated by this heterodimeric complex between UFD1 and MPL4. Interestingly enough, MPL4 doesn't bind to P97 in the absence of UFD1, and each member of the UFD1 MPL4 heterodimer is destabilized if you deplete the other. So this has been shown genetically. We observe no changes in VCP abundance, kind of consistent with this known biology, prompting us to speculate that maybe our agnotac directly targets UFD1 and MPL4 degradation, or somehow contributes to this destabilization. And so, though the degradation of NPL4 appeared to progress, as I mentioned before, equipotently in the same kinetics as UFD1, the levels were stored upon proteasome inhibition, but not by netylation inhibition. And in addition, the negative control analog with the methylated glutaramide and cereblon depletion both blocked degradation of both proteins, implying that the degradation of both proteins was dependent on cereblon and is likely not the result of agnotac binding alone, but only one is dependent upon sort of cereblon-based activity. And so we conducted SETSA binding assay and showed that we see stabilization of UFD1, but not MPL4, indicating UFD1 is the main target. In fact, we see a little bit of destabilization of MPL4. And so, sort of long story short, what we think is happening is our agnotac is recruiting cereblon to UFD1, causing degradation of UFD1 through ubiquitination and proteasomal degradation. But that recruitment of cereblon disrupts a protein-protein interaction and causes MPL4 to be degraded through its sort of canonical endogenous pathways, which aren't really known. I wanted to share one more story of how we can use these agnotacs right from our screen to perform functional studies of proteins that currently lack useful chemical probes. And as I mentioned earlier, we identified a relatively selective degrader for this enzyme called PLOD2. PLOD2 catalyzes lysohydroxylation of collagen peptides, and it's required for the formation of stabilized collagen crosslinks, and it's critical for EMT and migration and metastasis of various cancers, and so it's overexpressed in many cancers, and it's also needed for tissue repair and fibrosis. Surprisingly, there's actually no selective inhibitors for PLOD2, giving us an opportunity to use this first-in-class PROTAC to see whether or not if we're perturbing PLOD2 function. And so the DC50 is around 23, 25 micromolar. We don't get complete degradation, but quite significant. It's rescuable by netylation and proteasome inhibition. We have SETSA target engagement telling us that we're actually binding PLOD2. And so we subsequently observed that if we treat MDA MB231 cells with our compound and perform this wound healing assay or scratch assay, We see agnotac-treated cells had substantially decreased migration rates compared to the DMSO or the glutaramide negative control, the one that won't bind cereblon, indicating to us that that if you degrade PLOD2 in this context, you can reduce these sort of cancer migratory patterns. And so I'll just conclude by sort of summarizing and hoping that I've been able to convince you of the utility of our photo affinity platform to identify ligandable proteins, but also sites on proteins on functionally diverse proteins throughout the proteome, and that these can be used as pocket probes to identify first-in-class chemical probes and inhibitors for challenging protein targets. Very often, we uncover sort of these interesting mechanisms of actions. We uncover glue mechanisms, we uncover classical inhibitors and degraders as a story as related to 1504 that I discussed today. And on the agnotech side, we feel that we're sort of providing first principles of how chemoproteomic-based ligand discovery and target discovery can perhaps be translated to more function-focused libraries to yield first-in-class degraders for proteins. I think these agnotechs right off the screen are not super potent, but very often they're first-in-class chemical probes and could be valuable starting points. You can imagine using photo affinity probes or other methods to map the binding site and kind of using the template laid out to optimize the scaffold. Since the screen doesn't give you functional information right off, we're interested in integrating with other profiling strategies like ubiquitogenomics to tell us which targets might be direct targets as opposed to indirect targets. We're of course expanding the libraries in other directions as we've done with our chemoproteomic libraries. So with that, I just wanted to thank the folks that did the work. The SLC 1584 story was really led by a graduate student, Stephen, in our lab, along with a graduate student, Daniel, in John Tajaro's lab, along with Pazzo. Jacob and Wei Chao really have innovated our binding site mapping studies. And Ines Forrest, a graduate student in our lab, well, she just defended, really led the efforts of the agnotech story in collaboration with really fantastic collaborators at AbbVie. So with that, I'd like to thank them and thank our funding sources, and I'd be happy to answer any questions.